So what is the function of kidneys, or what is the function of the nephrons? Nephrons are the ones which does the actual function. Okay, how much our blood passes through the kidney or the nephron, the blood is getting purified or filtered of its nitrogenous waste. Okay, now there are 3 steps in the formation of urine. Now what is the nitrogenous waste in the form of produced in our body or by us in the form of urine? Okay, now the formation of urine takes place in the following steps. The first step is filtration, which is also called as ultrafiltration, because almost whole of the blood which enters the kidneys is getting filtered, and almost all the things except the blood cells and the proteins of the plasma. Blood cells and the proteins do not enter the filtrate. Okay, like all the others except these 3 RBCs come out of the, is present in the filtrate. Okay, that's why it is called as ultrafiltrate. Nearly 1000, 100, 1200 ml of blood is getting filtered every day. Per minute. But only, we pass only 1.5 or 1.5 liters per day. So what happens to the remaining of the filtrate? Where does it go? What happens to that? Most of the things, most of the water from this filtrate, now it's not called as urine, children, it is called the filtrate, right? Now from the filtrate, most of the substances and most water is reabsorbed. Even urea is also reabsorbed. To maintain, when we talk about excretion, we have to talk about osmoregularity also. When we, any organisms lose water from the body, from the cells, finally from our cells, what happens? The cell become, the cell's osmolarity increases. Osmolarity means the salt content increases. Okay, to maintain the salt balance, the correct salt balance, water is either absorbed or it is given out, exosmosis, endosmosis, all that you know that. Okay, that's why we feel thirsty. When our body loses excess water from the body, we feel thirsty. We drink water. Okay, so our body knows to maintain this loss of water and everything. To maintain osmolarity, our organisms retain some of the urea or uric acid formed from the filtrate, right? So most of the things are reabsorbed. Okay, and finally when you come to the distal convoluted tubule, whatever should come in the urine is finally secreted. Mainly urea is there, water is there, plus many other substances which has to come into this excretory waste will be secreted into the distal convoluted tubule, into the collecting duct. Okay, like our diffusing membrane of the alveoli, from where oxygen diffuses into the blood. Same way here, from the blood, the blood has to get filtered into the Bowman's capsule from the tuft of capillaries. Okay, now the blood has to pass through, or the filtrate has to pass through mainly 3 regions or 3 membranes or 3 layers, which are the endothelium of the glomerulus blood vessel. Endothelium, which is the epithelium, all are epithelial cells, right? Endothelium of glomerular blood vessel, and epithelium of the Bowman's capsule, and the basement membrane between these 2 things, these 2 cells. Okay, epithelial cells is present on a basement membrane. Yes or no? Right. So epithelium of the glomeruli and epithelium of the blood vessel, these 2 layers plus 1 basement membrane between these 2. Okay, and also these Bowman's epithelial cells of the Bowman's capsule are arranged in a way such that they leave slits. See the slits here shown here, the slit like things, right? which serve as a sieve, which serve as filter for the blood to pass into the Bowman's capsule. Okay, so these are called as podocytes. The epithelial cells of the Bowman's capsule leaves gaps or slits, filtration slits, called as podocytes. Okay. So as I told you, almost all constituents of blood plasma except the proteins and also the blood cells do not pass into the lumen of the Bowman's capsule. So nearly about 1100 or 1200 ml of blood is filtered per minute. Okay, right. And the amount of glomerular filtrate formed per minute is called as glomerular filtration rate, GFR. What is GFR, children? It is glomerular filtration rate, which is the amount of glomerular filtrate, not urine now, it is the amount of glomerular filtrate formed per minute. Okay, normal GFR is 125 ml per minute. Okay, so nearly 180 liters or 125 ml per minute is formed actually. So 180 liters of glomerular filtrate is produced daily. But 99% of this is reabsorbed by the renal tubules. Almost all regions, especially the Henle's loop, takes great participation in reabsorbing almost all regions, children. Okay, so reabsorption takes place at different stages according to concentration difference. These are the areas, the shown areas. This is one nephron shown here with all those parts. These are areas are this is the cortex and this is middle, of course. Okay, these spaces, all these spaces are the interstitial spaces. Interstitial space we call, right? So this, from the interstitial space, things may be getting absorbed into the renal tubules, things may go out of the renal tubules from anywhere. Okay, this arrow mark going out, arrow mark going inside, all these shows whatever is given out into the kidney tissues, and wherever whatever is absorbed into the renal tubules also is shown here. Okay, so from the filtrate, so 99% of this is reabsorbed. 99% of this is reabsorbed by the renal tubules. So normal volume of urine released is nearly around 1.5 liters. 1200 or 1200 liters of blood is filtered per minute, but during a day we release only 1.5 liters per day, right? Okay, so what are the substances reabsorbed? Glucose, amino acids, sodium ions, water molecules, urea, and many other components are reabsorbed into the kidney tissues. These represent, I just now showed you, these represents the kidney tissues or interstitial space. Okay, so there may be passive absorption, active absorption. Active absorption involves energy for absorbing. Passive means just passively according to gradient, difference in gradient, the things move in and out of the tubules. Okay. So most of the absorption happen in the PCT, which is the proximal convoluted tubule. Okay, 70 to 80% of electrolytes means ions, and water is reabsorbed from the PCT. And loop of Henle's maintain high osmolarity of the medullary interstitial fluid. The loop of Henle is present in the medulla region, that, juxtamedullary nephron, right? So by the time as the filtrate is passing through the Henle's loop, most of the substances are reabsorbed into the kidney tissues. And the osmolarity, the salt content of the medulla increases. It gets increased. Okay. And descending limb is permeable to, which is descending limb, which is going down, descending limb, and the other one is the ascending limb. The descending limb is permeable to water but almost impermeable to electrolyte. Okay, and this concentrates the filtrate. The filtrate, as it is moving down the Henle's loop, the filtrate which is passing here becomes more and more concentrated as the water is getting reabsorbed into the kidneys. In the ascending limb, minimum reabsorption occurs. It is impermeable to water but allows transport of electrolytes. So the filtrate gets diluted. As it comes here, it gets again diluted. And in the distal convoluted tubules, only conditional reabsorption of sodium ions and water take place. And the collecting duct extends from cortex to inner parts of medulla. It reabsorbs, the last reabsorption happens in the collecting duct. Okay, so large amount of water and it will concentrate reabsorbed finally, because when we talk about excretion, we have to talk about conservation of water also. We have to excrete our waste with minimum amount of water. Okay, that is why most of the water from the urine is not called as urine, is reabsorbed. Okay, so the last part, the collecting duct, from the collecting duct, most of the water is reabsorbed and the urine becomes concentrated now. It also allows passage of small amounts of urea into medullary interstitium to maintain the osmolarity. Okay, children. Now, urine formation and counter current system. Now, what is this counter current system? Now, if you take the Henle's loop, the U-turn of the Henle's loop, one of the limb is descending, the other one is ascending. That is, the filtrate is descending in the descending limb, and in the other one it is ascending. Right, and also you know that this Henle's loop is supplied by a tight capillary network called the vasa recta. Right, so inside the kidneys there is millions of nephrons. Okay, and all these vasa recta and the Henle's loop, ascending limb, descending limb, all these are placed so very close together. Okay, and it forms, that is sometimes, not sometimes, always, the as just now I told, in one of the limbs the filtrate is descending, and the other limb it is ascending, and it is supplied by vasa recta, you know that, right? In that vasa recta, adjoining vasa recta, sometimes the the filtrate may be flowing in the opposite direction. So all these together forms a counter current system, counter current mechanism, which altogether or finally results in the formation of absorption, secretion, reabsorption. Everything happens. See, in this diagram it is shown, right? In this diagram it is shown, it is this is the medulla part, this is the cortex part. See the concentration, the osmolarity increases from 300 to 200 to 1200 osmolarity, that much, but because of reabsorption of from the filtrate, sodium ions, water ions, electrolytes, glucose and urea and many other substances are reabsorbed, and it increases the osmolarity of in the medulla region of the kidneys from the filtrate, from the Henle's loop and the vasa recta together forms a counter current system, and it helps in the formation of concentrated urine finally. Okay.